Dr. Rafael Pulido from the Biobizkaia Health Research Institute. The stand is yours.
Thanks a lot for the invitation, and it's a great pleasure to share our results and to see how the other researchers share the results with us. It's our first participation in this international conference, and I would like to keep participating in the coming months. So as Merce has mentioned, we are here a consortium of several labs working in Bilbao area on the CTNNB1 syndrome, and in close collaboration with the Spanish Association. And all the work that I'm going to mention today forms part of this work together work, but I'm going to focus on the work that we have made specifically in our lab in Biobizkaia, where we are located, which is basically molecular biology, cell biology work, very, very basic. So I have to let you know that what I'm going to mention here is nothing related with the patients. We haven't touched a cell from a patient yet. All the work we have made is work in vitro with recombinant material based on the genetic information on patients. And we think this is, let's say, a feasible approach to get very nice clues on what could happen with the patients in their real life. And I guess in the next future, we will be working with cells from the patients themselves. I'm going to give you a very, very short introduction on beta-catenin and CTNNB1. It has been mentioned already several times, and maybe the next talk by Sonia also she will mention, but I want to make a specific aspects to mention. Then I will mention the experimental in vitro result that we have obtained in our lab, and some ideas and conclusions that I would like to put on the room, especially to discuss later if this is possible. And there is also a particular thing I want to emphasize now. In the program, I don't know if in the final program is still Carmen Aguado from Ciberer Biobank. It was a talk by Carmen Aguado, which is the coordinator of the Spanish Biobank, which is collecting samples from the CTNNB1 patients. And she was supposed to talk at the end of this session, but she's not going to talk. And I'm going to present at the end of my talk a bit of a view of the Spanish Biobank that is collecting samples from the patients, CTNNB1 patients. Sorry. Just to make a point on two very important aspects on the beta-catenin CTNNB1 function that I think is highly related with the disease that the patients have. As it has been mentioned before, beta-catenin has two major functions, which are as a transcription factor. This is I'm going to show some functional results on this. Ninja also shows our results. This is the major function, which is more studied for beta-catenin, but this protein also, it's playing a critical role in the intercommunication between cells. And these are two highly independent functions of the cell. Here we are going to discuss the function of beta-catenin mainly on the context of the transcription factor, but there is another function that maybe is more important in the disease than the transcription factor itself. This is just a warning to all of us. This is one aspect. The second aspect is that this gene is a very, very, very interesting gene from the research point of view, because it's associated with disease in a very, very particular way. It's highly associated with cancer, somatic cancer, with specific mutations in a very, very region of the gene, which causes high expression of the protein. This is very abundant to have, it's very frequent to have a cancer that has high expression of beta-catenin, and this is very, very well studied. Actually, most of the information we know about beta-catenin is because of its involvement in cancer. And actually, we went to work with this syndrome because of that, because we are a lab working historically in cancer. And this is completely different with what happened with the with the CTN syndrome patients. They don't have cancer, and they are not expected to have cancer, which is good news, but they have the syndrome. And the syndrome has a complete different pattern of mutations in the gene. If you have mutated the gene in a somatic tumor, the mutations go on a very particular spot in the gene, and in the syndrome, this doesn't happen. In the syndrome, the gene is touched in many different parts and mainly generating what we have seen already, truncated proteins because of nonsense mutation or frameshift mutation. So the gene is strictly regulated in vivo to avoid, let's say, these diseases. But we cannot forget about this dual function of beta-catenin and also thinking on the therapy. The major challenge on therapies for reconstituting beta-catenin function in patients is not causing cancer, because beta-catenin, if you have too much, this is risky to have cancer. So this is just what I wanted to highlight. This is the cohort we have been working with, and Mercy already mentioned. And just to highlight that we don't have missense mutations. Actually, my point of view is that these missense mutations are marginal in the disease, and even they are not as strongly related with the disease as most of the other mutations. We have the closer we have in our cohort to a missense mutation is in frame deletion of 3 residues, which is here indicated with this. I cannot point with one of these mutations, but we have, as you see, 8 nonsense, 10 frame shifts, 1 in frame deletion, 3 splicing, and 2 gene deletions. Most of them, by definition for the geneticians, are loss of function mutations, or very likely loss of function mutation, just because they generate truncated proteins. But in our opinion, this is not that simple, and that's why we have studied in detail from the molecular point of view at the protein and the functional level, this mutation in this cohort. Our idea is to complete this work that we have quite complete already with the Spanish cohort. We want to complete with the Portuguese cohort that we are also working in collaboration with, and we will have a full vision of the Iberian cohort of CTN-NB1 patients in the experiments we are making. We have been making experiments considering each patient is different. Each patient is completely different from the molecular point of view. From a genetic point of view, many of these patients are clustered together because they all have a mutation in the same gene, which generates a truncated protein. And then we have analyzed these potential truncated proteins one by one. And as you can see in this image here, we have been using antibodies against beta-catenin that recognize different parts of the protein to be able to study the protein well. And as you can see, the different mutations. And actually, Laura, now I see that when you have put the slide in your PowerPoint, this change. Okay, it doesn't matter because the mutations itself are not important. What I want to highlight is that all these bands that you can see in these Western blots indicate the expression in our in vitro systems of the truncated proteins that likely or that possibly are present in the patients. As you know, all the patients with this syndrome have 1 wild-type allele and 1 mutated allele, and then they are in their body a combination of proteins, which is reflected by this image. As you can see, the size of the proteins can be very different. And then what we have made with this is to test the functional activity of the truncated proteins, as Ninja was saying before, with the, I'm sorry, Ninja. This have also happened to me with the names in Euskera, that I always make a mistake because I'm not from here, and then the names from people from the East. Nina. I'm sorry, Nina. Sorry. Well, you have to call me Rafael, which is also funny. As you can see, most of the proteins that come from our core are partial or total loss of function for beta-catenin transcriptional activity, with the exception of this, the one in the bottom here, that has quite good activity and is the deletion of 3 residues. It could be quite similar to a missense mutation. But also I want to highlight that we have detected a group of proteins that actually are truncated proteins, but that have what we call residual transcription factor activity. And we're investigating this because we think that this might be relevant in the disease. We see mutations that are completely absent function for this function, but we see some mutations that have some, and we don't really understand why, but we think this is important. And also an observation that from this set of experiments, for us, very interesting, was that in spite of being generating highly truncated proteins, which by definition should be kind of unstable, even don't exist, we see very well in our gels and in our experiments. And this was telling to us that maybe this could be something that could really exist in the patients. The patients could have this protein that you can see in the slide. So what we have made with this is to test what we call the protein stability of the truncations, meaning how they, if I can, meaning how they behave in the cell in terms of how long they are alive, the proteins. And this has been very interesting because we have been able to make two major groups of proteins that come from two major groups of variants in this disease. Some proteins or some variants have a stability, or they like to be in the cell more or less like the normal allele, like the beta-catenin normal, and some of them don't like at all, and it's very difficult to detect, more difficult to detect in the experiment. And this, we have made like two groups of proteins, which here are indicated by two colors, green and red. The red color indicates proteins that don't like to be living inside the cell. The green ones, they like to be, and you can detect. I don't know if this is kind of understandable, but you have, at least we have been able to make two within our cohort, which is quite, let's say, common, the mutations we find. We make two groups that we think biochemically they behave different. And then we have been testing this, Merce actually have been testing very carefully to look for potential associations between these two groups of mutations, these two groups of variants, and phenotypic manifestation of the patients. And most of the, Merce maybe will comment later if there is a discussion on that, but most of the tests she has made, we haven't found any association, is as expected, because here the heterogeneity is that big and the cohorts are that small that it's difficult to find association. But interestingly enough, we have found that with some cognitive tests that Merce has been checking, we see a highly significant difference between the group of variants that are more stable in the cell and the group of variants that are less stable, as if indicating that there are variants that could predict a better behavior in terms of performance for some of these cognitive tests. This is a bit, let's say, very preliminary because of the small number of samples, and also because we would need to have this double check with another cohorts. But at least we have been able to detect this difference that we find of interest. And from the clinical point of view, could have a value. And then we are also making in the lab some experiments, always in the lab, always in vitro, trying to use the idea of how to reconstitute the expression or increase the beta-catenin expression levels in patients, because this could help, independently of any gene therapy, this could help the life of a patient if he could modify a bit the levels of beta-catenin without getting into cancer, or if he could be able to rescue some of the alleles. And we are making this with the two approaches that I think at least one of them some other groups are making, that is trying to increase the beta-catenin level expressions of the normal allele to see if this could compensate for the loss of the mutated one. And we are also making experiments specifically addressed to the patients with nonsense mutations in the gene. These are also preliminary experiments, but they are promising in the way that we can use compounds that to certain patients that have certain stop codon mutation, we can reconstitute partially the expression of the full-length protein. So this means that potentially this child, this person, could get a benefit of a drug that would rescue a bit the mutation, the variant that they have. As the conclusions, I want just kind of to read to you, just to discuss later. if I can read, because now they have changed. Laura. Well, as some conclusions, that's why I make a PDF. No. I make a PDF, but Laura changed the PDF to something else. I always like to put PDFs because then things are not changed. Well, okay, some idea that I think many times people don't have these ideas, and I think for research, you need ideas. I do need ideas, so I really like that people, please give me ideas. I don't know what to do, no? So, truncated beta-catenin variants might be present in patients. This is nothing that I have read nowhere. Like, no, they have stop codon and there are no proteins. Maybe they can exist, and maybe they can have a bit of transcriptional activity or a bit of function. This needs to be checked carefully. Of course, we haven't found clear genotype-phenotype association between the genetic changes we have tested and the phenotypes, with the exception of some cognitive tests that we could associate with the stability or the size of the truncated protein. Just thinking on groups of patients that have truncated proteins, because then we have all the group with missense mutations that have no truncated proteins. And then finally, the inhibition of the degradation of the beta-catenin or the increase of the synthesis of beta-catenin could help patients, and also the reconstitution by translational read-through of the patients with nonsense mutations. Our work, which is ongoing, how am I on time, Anna? 1 minute. And then I have 15 more minutes talking because of Carmen. No?
We're gonna have to shorten Carmen to the main slide, the main message for Carmen. Sorry for Carmen. Otherwise, sorry for Carmen.
Our current and future work is with the work that Carmen is making, collecting with the patient, and the families from the association, which are donating samples to the Xavier Biobank. We are planning to work with fibroblasts and to perform experiments with cells from the patients that have to corroborate many of our in vitro experiments. It's for us especially interesting all the aspects related with the missense variants in this disease, missense variants of patients which we don't have in our cohort because they are infrequent, but some of the results, they have been tested already, and we have also been making results analysis of missense variants. In particular, our perception, this is something that it will be discussed. Our perception is that gain of function variants in beta-catenin are not likely associated with the disease. This is our perception. And this we have experiments and okay. And then finally, we would like to explore the possibility to specific patients with specific nonsense mutations being treated with highly precise drugs that help them to reconstitute the mutated allele. This is something that we are also actively working. And then if we go, these are the agencies that are currently funding our research, and of course in combination always with the association. And then I will mention very 15 minutes talk.
No, you can take maximum 5. Maximum 5. 5. Go.
We started, we belong to a network in Spain that is called Xavier, that belongs to a bigger network, which is a Xavier, which are labs distributed in all Spain working in specialized areas. And there is one Xavier called Xavier, which is specialized in rare diseases. Here in this map, you have many different labs in many different, you will see, I just put this on purpose to see a map of Spain. Of course, we have there up in Bilbao, and the Biobank Xavier is in Valencia, east, south Barcelona. And then what they have started to do is, in a, let's say, a highly regulated, high quality level, collect samples from the families to have a collection of samples that can be used to do further analysis, genetic analysis or experiment with the fibroblasts or whatever the researchers would like to do. This is, as in other biobanks, highly regulated by the hospitals with the consent of the patients. And then the important thing is at the end, the Xavier biobank is going to have a collection of blood and a collection of blood and cells that come from the skin biopsy that some of the children need to take. This is tricky for the families to get this biopsy. Even for healthy donors, it's not nothing nice. But at least we will be able to have, the researchers will be able to have, and this theoretically will be available for everyone. You just have to request it appropriately to the biobank. And then I guess with this sample, we will be able to work to do quite important discoveries on the disease. And I think this is what they have now. Here you have in this slide the address of the biobank if you want to contact. Carmen Aguado is the person in charge. And thank you for these 5 extra minutes. Thank you very much.